All right, we're finally up and running, 3 minutes late, but we'll make it work, right? All right, guys. Hey, we're starting. Except the keyboard's not working. Anyway, or the PowerPoint, or the projector. Nothing's working for me today. Roll, same as always, but because there's only like 25 or so of us here. Front is 8:30, back is 9:30. Just trying to waste less paper. And we'll get that up. The first one's a reference to Dr. Mott's information that goldfish make essentially alcohol. You guys remember probably 2 weeks ago I told you guys he'd bring up the goldfish, and that's something to pay attention to. Whenever he has like his fun little tidbits, mitochondria are maternal only, goldfish make ethanol, stuff like that, that's something to remember because he likes to turn those into test questions, right? Just because, I don't know, it's a fun fact about biology. Dr. Mott loves biology, so he's going to put that on his test. So I don't guarantee it or anything, but it's also not that hard to remember, so try and remember it. All right. Our biology fact for today, there are 8 times as many atoms in a teaspoonful of water as there are teaspoonfuls of water in the Atlantic Ocean. So you think how big the ocean is, right? How many teaspoons are in the ocean has got to be just some number that you can't even calculate, right? And in just one of those teaspoons, there's still more atoms in there than the number of teaspoons making up the whole ocean. So that just shows you how insanely small these atoms are and how insanely packed they are, right? So I just thought that one was kind of cool. Not technically like biology, but it's science, it all works out in the end. Our study tip is to teach it to someone else. It challenges you to actually know what you're talking about well enough to explain it to someone else, right? So for me to have to get up here and explain what we learn in lecture to you guys requires that I actually kind of know what I'm talking about. You guys were here last time. Wasn't perfect. But if you sit down with like a roommate or a boyfriend or a girlfriend or whoever, and you explain the citric acid cycle to them, right? You have to know it well enough to explain it to them, right? If you know it well enough to teach it to someone else, you can be pretty darn confident that you know it well enough to answer test questions on it, right? So, I do this all the time for all kinds of classes. I find someone else in the class or someone who already took the class or something like that, and it doesn't even have to be like me teaching them, I don't say sit down, I'm going to teach you about chemistry, I just say like, hey, let's talk about this topic, and we just go back and forth about it, and every time it helps me learn it so much better than just listening to someone else say it to me and then assuming that I'm going to remember it 4 weeks from now on the test, right? So try it, teach it to someone else. It will work, I promise. Okay, so because I can't talk about Exam 1 material, I always hesitate to ask if there are questions because someone always asks an Exam 1 question. But do you guys have questions about non-exam stuff before I just jump into slides? Oh, you faked me out. No?
You may cover this today, but I'm having a hard time conceptualizing why we need to move electrons within the citric acid cycle and why that's so important to this whole system.
You're talking about mostly like in the electron transport chain? Okay, we'll definitely talk about that. Other questions? This is a lot less fun with 30 of you. Or you 30 are just lame. No? All right, then we're just going to keep going. Aerobic respiration then consists of 3 main pieces, right? Glycolysis, citric acid cycle, and the electron transport chain. What is the whole purpose of aerobic respiration? Produce ATP. That's it. Like that's really why we run this whole process. Okay, we get like a few extra byproducts and whatnot, but we're doing it to get ATP, right? What's ATP for? Energy. This is not hard, guys. Come on. It's like pulling teeth with you people. It's how we run all of our processes, right? It is the electricity of our body, right? Everything runs on ATP. It's not actually electricity. I hope that wasn't confusing. It's just everything runs on electricity, right? In human bodies, everything runs on ATP, or can somehow go back to ATP, right? You get what I'm saying. All right, so this is what ATP is, adenosine triphosphate. It's an adenosine and 3 phosphates, right? And then ADP is adenosine diphosphate, adenosine and 2 phosphates, right? So what this is showing us here, I can draw. I always forget. I can actually draw on this computer. We've got an ADP and then an inorganic phosphate, just meaning there's no carbons on it, it's just a phosphate. And you stick those two together, you get ATP. Same idea with NAD plus to get NADH. It's nicotinamide adenine dinucleotide, right? And then you stick 2 electrons on it and you get NADH, right?
So that one, where do those electrons come from?
So if you guys remember ions? What are ions? Charged particles, charged atoms, right? So what does this plus on the NAD mean? It's got a positive charge to it, right? Now, what charge do electrons have? Negative. How many negatives am I having? 2. If I take 2 negatives and 1 positive, what do I get? Negative. How many? 1, right? -2 plus +1 is -1. But so why doesn't this NADH say NADH minus? Because of the hydrogen. Hydrogens are 1 proton, 1 neutron, 0 electrons. In this case, this is considered a bare proton, a bare hydrogen. So this positively charged hydrogen joins up, and we end up with a neutral charge. These 2 electrons, it doesn't really matter for this class where they come from. They're just around, and we'll kind of see what happens there. But we take these 2, we stick it on a positive, we end up with -1. But by laws of nature, a -1 is going to attract a +1, and you're going to end up with neutral NADH. Okay? Making sense? Other questions?
So I understand the difference between oxidized and reduced when it's laid out separate, but what I have a hard time with is when it gets into the citric acid cycle, telling them apart.
Right. Yeah. That is the trickier part, and I'll kind of show you how that works. But let's. So I mean, we can always use this as an example, and then when we're going through the cycle, we'll use it for more examples. So what is this process here? Oxidation or reduction?
If it's going from NADH to NAD plus?
No. So I'm saying we take the 2 electrons and we stick them onto an NAD plus. So we're making an NADH. Reduction. You all hesitated because I said it hesitantly. You're right. I just like to mess with you guys. Why?
Yeah, because reduction is the adding of electrons on something, reducing its charge.
Perfect. I think that's actually my next slide here. Yep. So oxidation and reduction. Oxidation is to lose electrons. Reduction is to gain electrons. There are 2 mnemonics, acronyms, however you want to consider them, that I've learned. One is oil rig, okay? Oxidation is loss, reduction is gain. This is always with electrons, right? Don't forget the key part being electrons, okay? It has to be electrons. If it's protons, it's not it's neither, it's not a thing. So oxidation is loss, reduction is gain. Or there's Leo the lion goes GR, right? Lose electrons is oxidation, gain electrons is reduction. Whichever one works for you. If neither of them work for you, make up your own. If you can't come up with your own, memorize it for what it is. Do whatever makes sense to you, right? Oxidation is loss, reduction is gain, okay? So, I hate that the keyboard desynced. We're adding electrons to that NAD+. It is gaining electrons, therefore it's a reduction, right? So again, NAD+ is the oxidized form, NADH is the reduced form, because they're relative to each other, right? So if one is the reduced form, the other has to be the oxidized form. Does that make sense? Okay. So when we actually get to the citric acid cycle, I'll show you how to pick out of that and decide what it is. But I just wanted to give you the basics. There's that. Anybody have questions on that so far? If you don't say anything, I will just continue. Come back.
So is ATP and ADP, do they go through oxidized and reduction reactions against the phosphate? Inorganic phosphate. When ATP gains the third inorganic phosphate, is it gaining or losing electrons as a reduction reaction?
I don't think so.
Okay, good deal.
Because it's an inorganic phosphate and it's uncharged.
Okay, good deal.
So no, as far as I'm aware, that's not an oxidation reduction reaction. Other questions? You can always Google it and prove me wrong if you want. I may very well, but I'm fairly certain that it's not. No? Okay, so the first step, glycolysis. Where does it occur? Cytoplasm. Not in the mitochondrion, okay? I know at least 15 of you are going to get that wrong on the test, because we talk about glycolysis for 2 minutes and then everything else is in the mitochondria. So you spend so much time paying attention to the mitochondria that when you come back to a glycolysis question, you're like, yeah, it's in the mitochondria, like everything else. It's not, okay? If this is our mitochondria, they're kind of kidney bean shaped. Glycolysis is right outside of it, and then everything else happens inside, right? Kind of like how we see in this picture. Glycolysis is right outside, but citric acid cycle, electron transport chain, all that is inside, okay? So pay attention to where each of these occur, because that's definitely a test question worthy topic, right? He will probably ask you where at least one of these happen. Glycolysis is cytoplasm, right outside the mitochondria. So we put in 1 glucose, which are made of 6 carbons, and 2 ATP, right? Because we have to have energy to run these reactions. We get 2 pyruvate, which you don't really need to know that much about it, except that they're 3 carbon molecules, right? So we broke 6 carbons in half. Logically, we get 3 carbons and all kinds of extra stuff on top of it that makes it a pyruvate, right? We get 4 ATP out of it, so we net 2, right? Is anyone confused on what that means when I say we net 2? All right, if you're too embarrassed to say, you can ask me after or whatever. It just means 4 minus 2 is 2, right? We put in 2, we get out 4, so really we are left overall with 2. And we get 2 NADHs out of it, right? So that's what this looks like. I really like this super simplified picture here. It works for me in my head. If you don't like it, you can always find other pictures of it. The other thing that will help you memorize is to draw, okay? We're going to do some drawing today. And none of you look excited. Just take 30 seconds to draw that I've kind of boxed off in, I assume, red there. Okay, just draw it real quick.
Can you go back to the previous slide?
You want to see that one? And I have these posted, so if you're ever like, oh crap, I forgot to write something down, I'll go back. I don't have a problem with it. But if like after the session you're like, oh, I forgot what he said about that their slides are posted, it's recorded on Panopto. There's always extra chances. It's a lot of work, isn't it? You really have to actually try to care about your education. It's the worst. You can't just play Call of Duty every day. I don't even like Call of Duty. All right, take 30 seconds, unless you already took your 30 seconds. Just draw that real quick. Just go like this. Boom, boom, carbons. So do 6 carbon glucose, gives you 2 3-carbon pyruvates. Make sure you get your 4 ATP, your 2 NADHs, and your 2 ATP. Okay, raise your hand if you're not done. You still need a second to look at it. Okay. Take your time, but not too long because we're on a time crunch. All right, now, without looking at your notes, draw it again and see if you actually remember each portion. Did anybody not get the roll? Raise your hand if you didn't get the roll. Just you guys up front. Okay. Oh, and back there. You may just have to come up to the front after and sign it. I'm sorry. Unless you guys want to be good Samaritans, pass it all the way back. Do your thing. So draw from memory, just it's 4 pieces, 2 arrows. See how well you actually remember what you just did there. Got 5 seconds. 3, 2, 1. Okay, so now go ahead and check it. How close were you? This is just to give you guys an idea of, first of all, the idea that drawing stuff helps a lot in classes like this, where you have to memorize a lot of pieces of cycles and processes and all that stuff, right? If you can draw it from memory, you will do so much better on the test, right? Because then every time it asks you, how many NADHs are made in the citric acid cycle, instead of trying to remember and add them up and do all this stuff, if you've memorized how to draw that whole citric acid cycle, sit down, draw the citric acid cycle, then start the test, and then every citric acid cycle question that comes up, you can cheat, it's not really cheating, off your drawing that you just did on your scratch paper, right? And I know this sounds like something that I tell you in the session that you should do, and then you go home and you never do it. You really should. That is what I did on exam 2, and I got a pretty decent score on exam 2, right? I was happy with the score that I got, and a big part of it was being able to draw these things, okay? So please don't make this the last time that you draw that. Draw it until you can do it from memory, like 3.5 weeks from now when you take exam 2, right? And I'll remind you guys probably like in the super session that you should still draw these, but draw these, okay? Questions on glycolysis? No? All right. Clicker question on it then. Which of the following is true? Glycolysis produces a net of 4 ATP and 2 NADH. NADH is the oxidized form of NAD+. ADP plus inorganic phosphate equals ATP is an exergonic reaction. Glycolysis occurs in the mitochondria, or NAD+ has fewer electrons than NADH. Take a couple seconds to maybe work it out in your head. Who thinks it's A? B? C? D? How many of you are waiting because you think everyone else is going to say E? You're just waiting to see what everyone else says, huh? No, most of you probably knew. It's E, right? So why is it not A? It's a net of 2 ATP, right? So had this said 2, that absolutely would have been correct. Why not B? It's the reduced form, right? Because NADH picked up the electrons. Okay, so it gained, reduced is gain, right? ADP plus inorganic phosphate equals ATP is an exergonic reaction. Why not? Endergonic. Somebody remind me what an endergonic reaction is. requires energy input, right? And when you do an endergonic reaction, do you build or do you break? You build, right? You take two things and you build one thing. So if you ever see two on the left and one on the right, you know it's endergonic. It has to be, right? Why not D? It's in the cytoplasm, right? Now, how likely do you think it is that he'll ask a question like this on the test? Very likely. Most of you are saying very likely. Why? This was his clicker question. That's why. It's his question. Like this is the kind of stuff you'll see on the test. Okay, but you should be able to, hopefully, eventually, maybe not immediately, but as you go through the class, and the quicker the better, learn how to recognize if something is test question worthy. Look at it and go, is this too easy to be on a test? Is this way too complex of a question, way too deep of information, like half of what's on Mastering Bio? Is that just too much? Am I really going to see that on a test? Or is this kind of pretty spot on? Is this really something that he would ask? Right? And he loves, they're not trick questions, they're just barely wrong questions, right? Only one thing was wrong in each of them. But it's a good way for him to tell whether or not you really genuinely know it, or if you just have memorized a bunch of facts and can recognize trigger words like mitochondria and exergonic, and you're like, oh, it's got to be that, because I don't remember what exergonic is, but it's got to be that, right? So be able to tell what's a test-worthy question. That's my whole rant. Anybody confused on this and why it was? Okay, good, because we spent enough time on it. So the citric acid cycle. This thing's a B word, right? I kept it appropriate. I hope that's not offensive. I was just joking. I'm sorry, guys. I didn't love it when I took this class. I was like, I don't care. And it's so many things, and if you're in the same boat as me, you know, you may not care, and I don't care if you care, but you should care about the fact that this is a large piece of the chapter, and it's a complex process, therefore highly likely to see several test questions about the citric acid cycle, okay? So this is, again, where your drawing skills are going to come in handy, okay? You should be able to know that you take one 3-carbon pyruvate, you strip one of the carbons off, and you stick an oxygen on it, and you get CO2. You run an NAD plus next to it, it picks up the spare electrons. So that's where those 2 electrons were coming from that someone over here had asked about earlier. Those 2 electrons are coming from the pyruvate, okay, getting put onto the NADH. Then you should know you have 2 carbons, okay? You're going to put these into the actual cycle here, and you're going to have 4 circles. It literally does not matter what they are. They're called oxaloacetate, right? You don't really need to know what that is. If you want to know that it's called I can't even say it, then good. But just know that you take 2 carbons and stick it next to 4 other atoms, and it doesn't really matter what they are. And you just do the same process over and over, right? You strip off a carbon, it picks up 2 oxygens to become CO2. You run an NAD plus next to it, you pick up the spare electrons, right? Same thing, but now we're down to 1 carbon. So you push it down, you strip a carbon, you get CO2, you put an NAD plus next to it, you pick up the electrons. Always remember that there's 2 electrons involved here, right? It's 2 electrons, that's how we're getting NADH, right? We're running that reduction. So what would you then say CO2 is, oxidized or reduced? It's reduced, right? So this is where it's tricky, because you're like, well, I know that this is a reduction reaction, okay, because I know it's picking up the electrons here. But this CO2, is it getting oxidized or reduced? Someone's muttering oxidized. Anyone else brave enough to take a guess? Remember that they have to be relative to each other, so if this one is reduced, this one is oxidized. Holy crap. Should not be this hard. It is oxidized, right? Because what are we doing to this carbon? If we're giving 2 electrons to the NADH, what are we doing to the carbon? We're taking them away. So this is where people get tripped up. They go, well, the number of electrons went down. The number reduced. Didn't it get reduced? I don't know why they call it oxidation and reduction. Well, I know why they call it oxidation, right? Because that's an actual process. That's like how metals rust. That's oxidation, right? I don't know why it's called reduced. And you don't need to know why it's called reduced. If one of you knows why it's called reduced, share, but it's not a big deal. I guess you could consider it reducing the charge, like in the sense of more negative. Yeah, you could. Maybe that's why they call it that. I don't know. I probably should know. That's embarrassing. So don't. It's tricky not to get it backwards, right? Because you go, the number of electrons on carbon went down, so it reduced. No. Ask yourself, did it gain or lose electrons? Try to keep the word reduced out of it until you pick an answer. So say to yourself, did it gain or lose electrons? Okay? So this one lost electrons. It's the oil part of oil, or oil rig. So it's oxidation. Okay? I know it's tricky. It's easy to get it backwards. So hopefully this isn't the last time you look at oxidation and reduction, right? You have probably 3 or so weeks until the next test. Practice this a bunch of times. Okay? It's the only way you're going to definitely not get it backwards on the test. Because test comes, pressure's on, and we forget stuff all the time when we're under stress, when we're panicked, when there's a clock running, right? We're like, oh shoot, I got to do these fast. If you haven't practiced this a bunch, you're gonna, I hate to say you're going to. You very well might get these backwards, right? So practice it now so that when you actually have to get it right, you've done it so many times that you go, oh yeah, I lost electrons, I know it's an oxidation. It gained electrons, I know it's a reduction, right? Like that, okay? Anyway, that's my whole rant. We're back down here. We're taking another carbon away, reducing or oxidizing it. Oxidizing it. Okay, we're taking the electrons off. It's losing electrons because we're giving them to the NADH. So it is oxidizing or reducing? Reducing. Okay, so you may ask yourself, we're down here with these 4, but it's the same 4 over here. How is this, all of this, even a step? Shouldn't that just be gone? The idea is the citric acid cycle is very greedy, and it wants every single last bit of energy, every last electron stripped off of there, right? So even though there aren't any more carbons to take off, there are still some electrons, okay? So we can still add a few electrons. FAD plus is the same thing. I think it's flavin adenine dinucleotide. It's the same thing as NADH for our purposes. It's just flavin instead of nicotinamide or nicotine. I don't remember. You know what I'm saying?
So where did those 2 electrons come from?
Which 2?
So you said down here this, I don't even know, oxyl acetate.
This one?
Yeah. You said that one has 2 electrons right now, but where did those come from? Because we've stripped off all the carbon at this point.
Yeah, but these molecules, well, these atoms also have electrons.
Okay.
I don't know if these gray circles are carbons. Most likely they are, because almost everything is carbon. But these are carbons that still have electrons on them. So they still have a little bit of extra energy. So now we're just pulling the electrons off, without necessarily also pulling carbons off. Does that make sense?
So that top one in the loop, then, does that one have 2 electrons still, or has it been stripped off?
Right here,
Yeah.
it's at the end of its line. It's lost everything they can get out of it, and then we put it back into the cycle, and we give it more energy again by adding all those carbons and electrons and whatnot, and then we strip it back down. So that's why it's a cycle, right? We just run back and forth. Yeah?
Where does the COA come from?
This is an enzyme. It's for acetyl coenzyme A. It's just an enzyme. If you want to add it in there, it isn't a bad idea, but it literally just is from here to here. It just speeds. Enzymes do what? Speed up reactions. So just for this first little bit here, it's just making it go faster, and then it just goes back and does it again. So it kind of has its own little cycle up in that top right corner there. It's not like a super, well, it's important because the reaction would take much longer without it, but it's a small piece. Yeah, but that's just, it's acetyl coenzyme A. All right, so we keep stripping electrons, okay, and we use that energy to run an exergonic or endergonic reaction down here. Endergonic, because we built something. And then we oxidize or reduce that FAD+. We reduce it, right? We stuck electrons onto it. It gained electrons. Reduce is gain. We still have a little bit of energy left on these 4, so we're just going to do it again. We're going to add 2 electrons. So it's a reduction or oxidation. Reduction, okay? And then now we're down to the bare bones. We've run our cycle 1 time. How many pyruvates did we use? 1, okay? So this is where it can get tricky, because he can ask you how many NADHs are produced by the citric acid cycle per pyruvate. Go ahead and count them. Somebody said 4. 1, 2, 3, 4. Okay. How many are produced per glucose? 8. Why? Yeah. Per glucose, there's 6 carbons. We break that in half, and we get 2 pyruvates. So if we're getting 4 NADHs from 1, we have to get 8 from 2, right? So pay attention to that. I would be surprised if that was not a test question. Something along the lines of how many NADHs or how many FADHs or how many ATPs or something do we get from the citric acid cycle per glucose or pyruvate. So just know if it's per pyruvate, and this is where having your drawing comes in handy, right? Because you've already put this on paper, you can just go and count them up instead of trying to go, oh shoot, it was 4 or 8 or 2 or I don't remember. Right, if you've drawn this out, you already look at it. So if he says per pyruvate, just count up what's on your drawing. If he says per glucose, count up what's on your drawing and double it. Does that make sense? Do you guys see why that is? Yes? Do you have questions on the citric acid cycle before I move on?
Every time that carbon dioxide is produced, that's oxidation, right?
In this case, yes.
Right.
I don't want to say like all carbon dioxide reactions, but yes, all of these are an oxidation because they're having their electrons taken away and put onto that NADH. Well, onto the NAD plus to make NADH. Other questions?
Why is there not an oxidation reaction that takes place in that final step? Why is it just reduction? Because we had an oxidation down there with the ATP. How come what doesn't happen there? It's just the NADH.
Why are we not having an ATP right here? Is that what you're saying?
Yeah.
I don't know. My guess would be there's just not enough energy to put the 2 electrons on and make the ATP because we're running down to the end there. So if I had to guess, it's just out of energy, but I'm not sure. But that's also not something you need to know super well. But if I guessed, that would be what it is. And if I'm wrong, it doesn't matter because it's not going to be on the test. Okay, so I just preached to you about how important drawing is. Draw it. If you want to make it fancier than this, go ahead. If you want to make it more bare bones than this, go ahead. As long as you have, I would say, every time it does this, every time it does this, every time it does that or that, and I would keep track of your circles, okay? Other than that, you don't have to get like every single little piece in there, but those are the kinds of things you want to keep track of in the citric acid cycle. I'm going to give you 2 minutes because we're running out of class time. Class time, it's not a class. We're running out of session time. I'm your professor now, so I'm in charge. How much time did I say you have? 2 minutes? You get 1 minute. I changed my mind. I'm the professor. Plus, it's already been like 30 seconds, so I don't feel bad. All right, pencils down. No. If you haven't gotten every single little piece in there, you keep going while I talk. But the idea is eventually you should be able to draw this entire cycle in, I'd say, like 30 seconds. And you should be able to get every key piece in the right place with the right numbers, okay? Were you able to do that right now? No, right? It took a little more than a minute, about 2 minutes, right? And that's not terrible. If you have an hour to take a test and you take 2 minutes to draw a thing, it's not a huge loss of time. But obviously the faster the better, right? But the more accurate, the best, okay? So practice this. Tattoo it on your leg, tattoo it on your roommate's back so you see it every time. I don't know. Do whatever you got to do so that you have this memorized and so that you can draw it quickly and accurately, so that you can sit down on the test. And I'm telling you guys, I really hope you do this. Get into the test before you even open the laptop, before you do anything. And I really genuinely do this. I'm not just like saying you should do it so I sound good. Sit down, pull out your scratch paper, draw glycolysis, the citric acid cycle, and the electron transport chain. Boom. You should be able to do all 3 of those in like 2 minutes, tops. And then there will probably be 10 or 15 questions. Don't hold me to that, but 10 or 15 questions on all of those 3, and you can answer them like that because you can just look at your scratch paper, right? So please, it will help you if you can draw these quickly from memory before you even start the test. I really did that, I did. Okay, draw it again. Without looking at your old one. Do the best you can. You may miss a thing or two, but that's good, because it helps you realize which parts you already know well enough that you drew them from memory just the first time you tried, and it helps you see which parts you're a little bit fuzzy on. Maybe you missed an NADH or you missed a carbon somewhere, right? But see where you're lacking, see where you're already doing well, and then, I don't know, make this your nightly ritual, right? Every night before you go to bed, watch an episode of The Office and draw the citric acid cycle, and call it a night, right? Whatever your nightly ritual is, you're probably healthier than me. You like do some sit-ups and then go to bed. Do some sit-ups, draw the citric acid cycle, and go to bed. And studies have actually proven that learning something and then going to bed is actually a really good idea. Because during dreaming, that's actually when you consolidate your memories. So if you draw this and then go to bed, and then you wake up the next day, you'll have learned it a lot better. And I actually know that's true because in chemistry, I spent like 3 hours one day drawing all kinds of isomers and things like that. and I went to bed, and I literally dreamt about them. Like, I just had dreams about drawing these stupid chemical structures, right? But I got 100% on that chemistry test because I did it every night. I drew them, and then I went to bed. I dreamt about them, so I spent 8 hours still drawing them, and it was, it was for free. I didn't lose anything. I still slept, and I killed it, that test. I didn't get 100% on all the tests, but I got 100% on that one because I drew it and then went to bed. So I dare even one of you to try it and then tell me after the test if you dreamt about it. I want to know if I'm a weirdo. I want to know if that's like not actually psychologically healthy. No, it is. That's normal. That's what's supposed to happen. All right, running low on time, so go ahead and check your work here. If I distracted you with my talking, welcome to my SI sessions. That's just what I do. I'll have like midterm reviews, and someone will put that. They'll be like, don't go on so many tangents. And I'll be like, I can't. It's who I am as a person. It defines who I am. So check your work, see if you got all the stuff in the right places, see where you missed something, and make a note of that, right? Maybe say, I forgot to put an NADH here, or in a different color ink, add an NADH and have that kind of be like a note to you that that's where you forgot it. And if every time you check your work and you realize if you're missing something or not missing something, how well you're getting it, eventually you will end up being able to do this perfectly from memory, right? All right, questions on the citric acid cycle?
Do we need to know whatever the name of the first 4 carbons?
Oxaloacetate?
Yeah.
Not really. If you see that, recognize that it's talking about these, but I don't know, you don't really need to know how to like spell it or what it is or anything. Just recognize maybe what the name is.
Is there any difference between like the processing of NAD plus to NADH and FAD plus to FADH?
For the purposes of this class, no. It's flavin versus nicotinamide, but it's the same thing in the end for this class. I'm sure in microbiology or biochemistry or some super advanced class there is a really important difference. We don't care. I don't care. In this class, it's not important. That's a better way to say it. I'm going to get in trouble one day. I did once. I put out an announcement, and it wasn't even that bad. But I said, what's the sexiest enzyme? And the answer was helicase because it unzips your genes, which is what helicase does, right? It unwinds your chromosomes. And on my midterm, somebody was like, you need to stop with the sexual references in your announcements. And I was like, first of all, I even spelled genes, G-E-N-E-S, so it's not sexual. It's biologically correct. But also, it was one time. If you can't handle it, I don't know, watch the Panoptos or act like I don't exist. Hillary's probably nicer than me. Go to her session. I'm getting distracted again. So this is just a rundown. I don't want to spend a lot of time. This is what you get out of the citric acid cycle. Take a picture of it, draw it, download my notes, do whatever. But that just tells you what you're getting out of one citric acid cycle, right? So one pyruvate. And so if you have 2 pyruvates, then you got to double it. But this is one time, 1 pyruvate through 1 citric acid cycle. Okay? We're down to the wire here. Which of the following statements about the citric acid cycle is true? It occurs in the intermembrane space of the mitochondrion. The citric acid cycle produces 2 pyruvate from 1 glucose. NADH is oxidized in the citric acid cycle. The citric acid cycle reduces the carbons of pyruvate to CO2. None of the statements are true. Okay, we got to be quick on this one. So think about it, think about it, think about it. Who thinks it's A, B, C, D? I need to stop making these E so you stop getting nervous. E, the 6 of you who actually answered. Disrespect. Yes, none of these are true. Citric acid cycle, where does it happen? Matrix of the mitochondrion. Citric acid cycle produces 2 pyruvate from 1 glucose. Glycolysis does it, right? It's not the citric acid cycle. We do get 2 pyruvate from 1 glucose, but it's in glycolysis, right? And it's not in the mitochondrion, it's in the matrix. NADH is oxidized in the citric acid cycle. Reduced. And the citric acid cycle reduces the carbons of pyruvate to CO2. Okay. We're pretty much out of time. Maybe let's answer this. In the mitochondrial electron transport system, which is this, and you may not know this because we only heard it once in class, but just give it a try. Electrons are added to NADH. NADH is oxidized to NAD+. Protons are pumped from the intermembrane space into the matrix. The carbons of pyruvate are oxidized to CO2. ATP is consumed. Who thinks it's A? B? C? D? E? We had a little bit of a spread there. It's B. This is an oxidation reaction, right? Does everyone agree with that? Okay, because we're taking the electrons off to turn it back into NAD+. So why not A? Because of what I just said, right? We're taking them off, not putting them on. Why not C? That's later, right? That's not the electron transport system. The carbons of pyruvate are oxidized to CO2. That's earlier. That's in the citric acid cycle. ATP is consumed. No, ATP is created, right? Okay. Next session, I promise to go over all of these slides here. If you have questions about them, like if you're looking at them tonight or whatever, I'm always available via email or Canvas. Ask me any questions you want. I'm pretty good at answering pretty quickly, not necessarily correctly, but quickly. I always answer quickly. Got our roll down here, all right. If you guys